Welcome to The Explainer. Today we're diving into a fascinating story. It's about a scientist whose absolute love for RNA research actually pushed her to become one of the biggest critics of mRNA technology. So how on earth did her passion turn into dissent? Let's find out. In this quote right here from geneticist Dr. Alexandra Henrion-Caude really gets to the heart of it. You can feel this central conflict, right? Her deep appreciation for the RNA molecule is the very thing that drove her to speak out against what she saw as its misuse. So first, let's really get into where this passion comes from. If you want to understand why she started questioning this new technology so hard, you have to understand her entire career in RNA research. She has this amazing analogy to explain it. Think of DNA, she says, as this complete book of information, but it's just sitting on a library shelf, totally static. RNA, on the other hand, well, that's the action. It's the moment you open the book, you read it, you bring all that information to life. For her, that's where the magic is, in RNA's dynamic, responsive nature. Okay, so this love for the natural role of RNA flows right into one of her biggest arguments, that calling this technology a vaccine is, in her view, just plain wrong. And to really hammer this point home, she uses this clever bit of wordplay in French. So the official term is IRN messenger. That means messenger RNA. But she calls it RN menteur. It sounds almost exactly the same, but it translates to liar RNA. So for her, the crucial point is this fundamental difference in how they work. Traditional vaccines, she argues, they operate from outside the cell. They just send a signal to the immune system. But the mRNA platform, that's a whole different ballgame. It actually goes inside the cell and, as she puts it, hijacks your cell's machinery, basically turning it into a little protein factory. And this is where her objection gets really serious. She argues that if you turn a human cell into a factory for a foreign protein, you are, technically, making that person a genetically modified organism. It's a conclusion she admits is terrible to say, but one she feels is scientifically accurate and carries huge ethical weight. And this brings us to her specific, technical complaints. See, this isn't just some philosophical debate for her. She points to what she sees as very real problems with both the synthetic RNA being used and the system that delivers it. One of the biggest questions she keeps asking is, where's the off switch? Your body's natural RNA breaks down super quickly. But with this technology, she says the evidence shows that both the mRNA and the spike protein can stick around for months. There's just no clear way to tell the factory to shut down. Her first big technical issue is with the RNA molecule itself. See, they use a synthetic chemical letter in the code to make it last longer, but she argues this causes all sorts of problems. It can confuse the cell's machinery, causing it to misread the code and crank out faulty proteins. Plus, she says the cell actually starts to prefer this synthetic code, stealing resources from its normal, everyday jobs. Her second major concern? The delivery system. She calls the lipid nanoparticles a type of slime that helps the mRNA payload stick and spread everywhere. She raises concerns about their potential toxicity and even points to Pfizer's own studies, which showed these particles don't just stay in the arm. They travel to critical organs like the brain, the ovaries, and the testicles. Now, as you can imagine, raising these kinds of concerns came at a huge cost, both personally and professionally. Dr. Henrion-Caude talks about this incredibly fast journey from being a respected, sought-after expert to being a censored and attacked public figure. And this timeline just shows you how wild that shift was. She says she was first censored way back in February 2020, but get this, after that, a major French publisher actually chased her down to write a book on RNA. But when the book was released, the media backlash was so vicious that the publisher was apparently stunned and pulled the plug on her whole promotional tour. It got so bad that even her old mentor publicly wondered, I really don't know what happened to her. So all of these experiences, she explains, are what basically forced her out of the lab and into the streets. Her scientific critique morphed into a much broader fight for what she saw as fundamental freedoms. Her activism really boils down to these three things. First, that core medical principle, do no harm, which she felt was being completely ignored. Second, defending medical freedom, the idea that no one, especially healthy people, should be forced into a medical procedure. And third, she was passionate about preserving unity in a society she felt was being torn apart. And this quote became her signature, her rallying cry at demonstrations. She made a point of always starting her speeches by saying, vaccinated, non-vaccinated, we are one people. It was her direct attempt to heal the divide she saw being carved right through the middle of society.
So, you might be sitting there thinking, okay, but the pandemic's mostly behind us. Why does any of this still matter? Well, according to her, this whole debate is actually more urgent now than ever before.
And the reason is really simple. She points out that these mRNA manufacturing platforms are being built right now, all over the globe. This technology isn't fading away. It's being scaled up for whatever comes next. And she believes those fundamental safety questions still haven't been answered. And she is definitely not a lone voice crying in the wilderness. She's quick to highlight a call for an international moratorium, a complete halt on the mRNA platform. And she says it's been signed by over 2,500 medical scientists from more than 60 countries. That signals a pretty significant worldwide base of scientific concern.
When you take all this in, her perspective really leaves us with a massive question, one that goes way beyond just this one technology. It's a question for our time, really. In an age where medical innovation is moving at lightning speed, how do we, as a society, find that crucial balance between pushing for progress and ensuring we're taking every precaution?